Thanks everyone for joining us today for our information session on antibiotic resistant bacteria. We have a very special guest with us today, and I will let him introduce himself and tell you all the wonderful things he's done, and then we'll ask him some questions along the way too.
Awesome. Hello, everyone. My name is Ronald Bridgeborn. I hail from Buffalo, New York currently. I was once a student of the esteemed Dr. Matthew Deitch. Um, but I am a microbiologist by trade, but I did agriculture research. And under the mentorship of Dr. Deitch, I worked in soil, water, and ecosystem science. And I currently work for the EPA Region 6 in Dallas. um, as a community engagement environmental review division as an environmental scientist. And I also teach sustainability, um, intro to sustainability and perspectives in sustainability. And I'm grateful for the opportunity to talk about bacteria, poop bacteria, something I'm very interested in. And thank you guys for having me.
Thanks so much for joining us. We're excited to have you here.
Awesome. So for bandwidth, I will be taking off my video so that you don't see my head bobbing around, right? So today, greetings everyone again. Today we're diving into an issue that affects every one of us, which is antibiotic resistance, especially how it connects to our food systems. We often think of antibiotics as life-saving medicines that doctors use to fight infections. But what happens when the bacteria we're trying to kill actually learns how to survive those antibiotics? Now, that's what we call antimicrobial resistance, or AMR. So in today's session, we'll learn how AMR develops, how it spreads through our environment and food, and how scientists like you can detect it using PCR and gel electrophoresis, the same tools you'll get to use during your lab visit. Let's start with the basics, guys. What are bacteria? So bacteria are microscopic single-celled organisms. You might have learned that in class already. And they're everywhere. They're ubiquitous, which is a big fancy word for saying they're everywhere. They're in your soil, water, plants, animals, and even inside your body. In fact, you know, your body has more bacterial cells than human cells. Most bacteria are beneficial. How, you may ask? They help digest food, make vitamins, and recycle nutrients in ecosystems. But guess what? Out of all these millions of bacteria, a small percentage of them can cause diseases, right? So these harmful bacteria, we refer to them as pathogens. Some examples you might know, maybe from your classes, or might have saw somewhere, are E. coli, Salmonella, Listeria, and Staphylococcus. So these bacteria can contaminate your food and cause foodborne illnesses. Think of bacteria like people, right? Some are good citizens, but some are troublemakers. Antibiotic resistance happens when bacteria evolve to survive drugs designed to kill them. So here's a breakdown of how it sort of works. When, sorry, when we use antibiotics, most bacteria die, but a few survive. Because they are special genes that make them resistant, these survivors multiply and pass through resistant genes to offspring or even other bacteria. And so some common antibiotics you may have heard about are penicillin, a very popular one. And some other examples can be tetracycline, ampicillin, etc. Now, resistant bacteria they can carry genes like beta-lactam, tet A, which is tetracycline A, and other like methicillin. An example of an EMR is MRSA, which is an abbreviation for methicillin resistant Staphylococcus aureus, which is known as a major hospital superbug. So EMR evolution in action, basically bacteria are outsmarting us. So why should we care? Who really cares? So what, right? We often ask ourselves, why should we care about EMR? Why am I listening to this video? Now, the fact is, antibiotic resistance isn't just a medical issue, it's an environment and food system issue too. Did you know that every year, millions of people in the US get infected with resistance bacteria? Actually, it's 2.8 million, according to the CDC, and over 35,000 die globally. And so, in food systems, EMR threatens the safety of our food supply. And we all need food for energy and sustenance, right? Can you imagine a day without food? And so, with this happening, it increased the cost for farmers and reduces the effectiveness of antibiotics used in animal health. Now, if bacteria continues to resist our strongest drugs, even small cuts, surgeries, or infections could become life-threatening again. So, AMR isn't just about humans, it's actually a one health issue. And one health basically connects people, animals, and the environment. So, how does resistance, how does these AMR enter our food? So, farm animals, like the chickens we eat, the bacon we eat, the pigs, the beef we eat, the cows, are often given a lot of antibiotics, not only to treat illnesses and diseases, but also to help them grow faster and stay healthier in these crowded pens, etc. These crowded farming operations. So when these bacteria in these animals become resistant, they then can spread, right? And how can they be spread, you may ask? They can be spread through their poop. And guys, I'll be saying the word poop quite a bit. So they can be spread through the manure as fertilizer, which then contaminates soil and crops. And also during processing of them, you can contaminate meat and surfaces, etc. But also they can contaminate, they can flow into food systems through runoff where the water can carry bacteria into the rivers and lakes. So for example, Salmonella and Campylobacter are found in poultry and can resist multiple antibiotics such as tetracyclines, which I've mentioned. And so vegetables and fruits can also carry resistant bacteria if they are irrigated with that same runoff water I mentioned. So, as we can see, the food chain becomes a transmission route for resistant genes. So, as I've mentioned that, right? We've talked about how these AMR, how these antibiotics and so forth, and bacteria can be transported, right? What are the modes of transmission? What are different ways? So they can be spread through several pathways as listed here. One would be direct contact, which as the word states, can be done from handling, let's say, contaminated food or animals that have been contaminated, or direct contact with other people who have AMR. Also, indirect contact. For example, touching surfaces contaminated with it, tools, equipment contaminated with this resistant bacteria, etc. Another way is through foodborne transmission, eating undercooked meat, raw vegetables, and carrying bacteria. So it's very important to make sure you cook your meat and vegetables properly and thoroughly. Another one is via environmental transmission. And I would have mentioned runoff already via the manure or wastewater carrying resistance genes into rivers and soil. And lastly, I would mention airborne spread, which is basically the bacteria spreading via dust or particles being released during animal farming or from farms carrying these resistant microbes. So, as we can see, EMR doesn't stay in one place. It moves through ecosystems, water, and food webs. Think about this. Every time it rains near a farm, right there in Illinois, tiny amounts of resistance bacteria can flow into nearby streams and eventually into our food or drinking water. So antibiotic resistance, it affects everyone from hospitals to farms. And so without effective antibiotics, simple pathogen infections could become life-threatening. So on one hand, it's very important for human life, but on the other, it can affect us as well. From farm to fork. So let's visualize how EMR moves into our environment. Let's picture these animals here. When animal waste or fertilizers containing antibiotics are spread on soil, the bacterial resistance genes can then leach into our groundwater or surface water that we can either use for bathing in, for kayaking, or drinking. So another way is heavy rains or flooding can carry these bacteria downstream where they attach to sediments, etc. And we call this feed and transport. When antibiotics break down, the resistance genes can persist, and these are called ARGs, antibiotic resistance genes. So, once again, water, soil, and sediment become long-term reservoirs for AMR, cycling it back into the food chain, right? So this farm to fork AMR pathway, we can see where resistance bacteria develop. They can be spread via manure, water. They then go into the food chain. And then we as humans, we will ingest this food and then we are contaminated and the bacteria infects us. And then is subsequently disposed into the environment. So bacteria can contaminate the environment as well. So what are the main drivers of EMR, right? We see that social, environmental, economic factors increase infectious disease burden. And so we've seen that in the farming aspect, the overuse and misuse of antibiotics in animals and humans are a main contributor to these antibiotics being prevalent in our environments and food systems. So I would have mentioned this. So let's visualize this, how AMR can reach to our food systems. But I would just mention here bioaccumulation. And this is when the resistant bacteria can build up in aquatic animals. And then we can eat them. So let's say these resistant bacteria end up in our fishes or oysters, and then we ingest it. That's one way. And then another term I would like to notify is let you know about is remobilization, where flooding events re-release these buried bacteria and AMR into our food systems. These are some impacts and effects of AMR, right? It has impacts on our health, the economy, costly stays, agriculture, and so forth. We've mentioned the environmental impact. But we are concerned about food safety, right? So these AMRs, they contaminate our food or meat or produce or plants, and seafood supply. And when we ingest it, we develop resistance to that. As someone who, over the last 4 years, have battled a plethora of antibiotic resistance bacteria, I can tell you it's no fun. I've had at least 5 serious antibiotic resistance infections from MRSA to Staphylococcus, Pseudomonas aeruginosa, and so forth. And it's quite deadly. It can lead to one becoming septic and eventually becoming very ill. So it's very important as it relates to food safety. One thing we risk is the potential for new pandemics from resistance bacteria. So, excuse me, EMR in our food systems. How does resistance enter our food? Farm animals, right? As we can see, these farm animals are often given these antibiotics, not only to treat illness, but to get to grow faster, as I mentioned. And so it's then released into the environment. So livestock antibiotics, the overuse and abuse of this drives resistance. And so many bacteria that become resistant to them are E. coli, Salmonella, Campylobacter, and Pseudomonas, etc. And so another way is cross-contamination spreads bacteria during the processing or cooking. So let's talk about the science of detecting EMR. One way, let's talk about this exciting part. Now we've talked about how bad it is, so it's important for us to test it. And next week, you guys might be working on this. So PCR is an abbreviation for polymerase chain reaction. You may have heard this term before. So PCR is a technique that is used to amplify DNA, right? And so scientists use PCR to find and identify antibiotic resistance genes. Here's how it works, guys. First, you extract the DNA from your bacterial samples, which could be food, water, soil, even fecal matter and plant matter. You then add primers, secondly, and primers are basically short pieces of DNA that match resistance genes, right? And then the machine cycles through heating and cooling and then copies these genes millions and millions of times. So there are 3 stages here, I've briefly mentioned: denaturation, annealing, and extension, which is the DNA strands separate, basically. Then the primers adhere to the DNA strands. And then lastly, the Taq polymerase builds complementary strands. So it's a long process, but we get good results. And if the target gene is there, PCR makes enough copies for us to detect it. You'll hopefully get to see this be run or test yourself during your lab field trip next week. So scientists, excuse me, scientists test food, water, fecal, plant, all sorts of samples for AMR, right? And so that can be done via PCR, which we just mentioned. And so, as I said, the DNA is extracted and amplified, and the results appear as glowing bands when genes are found. So another method would be gel electrophoresis. And so once the DNA is amplified, we can use gel electrophoresis to see it. DNA samples are loaded into a gel, right? And so an electric current pulls the DNA fragments through. Smaller pieces move faster, forming distinct bands. So it basically lights up. So when you shine UV light on the gel, the bands glow. And these glowing bands tells us if antibiotic resistance genes are present. For example, A glowing band at a specific location might confirm that TET-A or any resistant gene is in your sample. It's like developing a photograph, basically. The image reveals the hidden story of the bacteria. So the DNA fragments travel through gel based on size. Smaller pieces move faster, larger ones move slower, and each band is equivalent to a specific gene, right? These are some steps here. And so we can state that each glowing band is like a fingerprint that helps you, as budding scientists, identify which resistance genes are present. So I've mentioned all these cool molecular tools, PCR, etc., that you and other scientists can use. But who else does this? The FDA tests food for resistance bacteria before it reaches stores. The EPA monitors water systems for antibiotics resistant genes. And hospitals and universities use them to track outbreaks and study resistance trends. And so they help us to answer key questions of food systems and samples. Do they contain bacteria that are resistant to these antibiotics? Where did the genes come from? And how are they spreading? So these techniques I've mentioned are the foundation of modern microbiology. And soon you'll be using them too. So what can we do as it relates to AMR and food systems? We can use antibiotics responsibly. We can discard of them responsibly by visiting your local pharmacies and discarding of them in those respective bins. We can practice good hygiene and cook your food properly and thoroughly. And we can support green sustainable farming. Thank you for your time, guys. And do you have any questions?
I was going to ask a couple. I don't, Matt and Russ, do you have any that you want to ask first, though?
No, you go ahead.
So thank you for sharing the part about how like the EPA monitors and things like that. Could you tell us a little bit about maybe like how often they would test a water sample or like how often they might test a food sample or something along those lines?
That's a good question. So it varies from region to region. In, in the Midwest, they would only sample during the warmer months, right? The EPA, that is the EPA, they would sample during the warmer months. However, city and state agencies within areas would monitor monitor systems monthly or biweekly. So it and then it varies from state to state and depends on their need. So, for examples, in areas such as Florida, the Texas, and so forth, that would have frequent flooding and rainfall and so forth, they do more intense sampling. But places such as Iowa, Nebraska, um, they would sample only in the warmer months. But the EPA sort of does their own sampling, but manages and assists city, county, and state agencies to monitor on a more frequent basis. And so their data is then sent to the agency or uploaded and sent for review, etc.
Excellent. Thank you. Matt or Russ?
I do have a question. Ronell, how common is it for state agencies to do or federal agencies to do these types of like this type of monitoring? Like, is this part of a routine thing or is this part of something that they would only do if there was a big problem that they detect someplace or that's reported?
And that's a good question. So, um, once again, it's situational dependent if it has been detected, but as of the over the last couple of years, Ohio Cincinnati lab, Cincinnati, the lab in Cincinnati has been partnering with NAMS, and they have been monitoring antibiotics, antibiotic resistance, bacteria, and ARGs, and fecal bacteria throughout the US. And they have a very big study going on right now. I think they have wrapped up their sampling and testing. So as of recently, this has become such a it's becoming more of a issue. And so there's a bigger push to monitor the ARBs and ARGs and so forth, because we are seeing how it's affecting everything from a One Health perspective. We as humans, or plants, or animals, or children, etc. So agencies are pushing for more frequent sampling and so forth. And there has been a big study recently been done. The data has not been published as yet. One of my colleagues actually is running that program, Dr. Franklin.
I do have one more question about food safety. Is AMR resistant bacteria the kind of thing that can be eliminated through like cooking food thoroughly, or is it something there's still some risk of? How risky is it just to eat meat in general compared to undercooked meat? I don't know.
And
Does that
that's a
make
good
sense?
question. That's a good question. Interestingly, I taught a lab, a techniques, sorry, a sanitation techniques lab, right? And so we demonstrated with food and samples how different cleaning agents worked. And so interest and different, the meat was cooked at different levels and temperatures, etc. And so what in this small lab exercise, we found that once your chicken is thoroughly cooked properly, right, you're safe, right? It eliminates and kills most bacteria. Majority of bacteria die at a certain temperature. Hence the reason it's very important to cook your food thoroughly at that respective temperature for that meat. So for example, pork and chicken would require certain temperatures. And once it reaches that temperature, it's almost 90%, 99.9% sure that all of that bacteria has been eliminated. Also, when cooking different meats, it's very important to sanitize your equipment that you're using, because touching meat to surface, fecal oral route, hand to mouth, the bacteria can easily be transferred. So it's very important to disinfect your workspace, be it cooking or handling meat, and to thoroughly cook your meat, because once you cook your meat, guys, I'm almost very positive and very sure that no bacteria or AMR will thrive. And parasites.
Russ, do you have one, or it's okay if you don't?
I do, and this might not be as educational as your presentation has been. So, is there like a set, like, don't eat leftover food after this long? Like, I just want like to end all the Thanksgiving arguments. Like, 7 days is probably for everything you got mold on it. Like, I'm just really kind of looking.
And that's actually a very interesting topic as well, because the fact of the matter depends on many things, right? So your refrigerator depends on what temperature your refrigerator and freezer is set at, right? And so typically, let's start with a non-meat, rice. Day-old rice, 2-day-old rice, you can get away with. Anytime after that, fungi and bacteria that's hard to see with the eye, it's too microscopic, grows. And therefore, you may have some bowel and diarrheal diseases that would arise to that. Coupled with that, different beans would ferment and so forth over time. And then let's get to the meat of it. Meat, right? Chicken, pork, duck, lamb, whatever you like, beef, etc., right? If it's stored at a certain temperature, right, it can last a good 5 days. If it's stored at a very cold temperature, it'll be good. If you put it in the freezer, even better, right? But it's very important. Storage of meat, storage and handling of meat is very important, cooked and uncooked, because even through handling cooked meat, said microbials can be passed on.
Ronald,
That's exactly what I needed to know. Thank you.
we know Russ. Thanksgiving's not too far away.
Right.
Ronell, if this one's too personal to answer, it's okay, but you mentioned you had you yourself were sick with some antibiotic resistant bacteria. Do you know how you came in contact with that? Or and then also like how did your doctors go about treating you?
Well, this might become some of a sad story, but whilst I was a student, a PhD student with Dr. Deitch, I contracted a bacterial infection called meningitis, Neisseria meningitis, right? And so that bacterial infection, I became septic and was admitted to the ICU. And for 4 years, I battled um, near-death experiences. I amputated, my fingertips were amputated, my feet were amputated. And with that, just as we've talked about this topic, I battled numerous antibiotic resistance bacteria. And so I would have been admitted to the hospital on 4 separate occasions for life-threatening ARB, same antibiotic resistance bacterial infections, such as the ones I mentioned, Pseudomonas aeruginosa, Staphylococcus aureus, E. coli, and Enterococcus facilis. And so I only know these names not because I'm a microbiologist, but I've personally been affected by them. And so it's easy to contract them. When you do have an open wound serves as a plate, a petri dish for anything to come on. So I would have sustained these from even water, right? From surfaces. And so it's at that stage, it's very important to be very clean, especially when you have cuts. And so so I would have had all these different infections, and they would have given a plethora of different antibiotics. And due to the overuse of antibiotics in these hospitals and my constant flux of being in and out of the hospital, I acquired hospital infections as well. And so Dr. Deitch would have been around, I would have been around him those days. So he knows about that. So it's easy to contract these things, especially when you're immunocompromised or even ill, because it makes you more susceptible to those things.
Thank you so much for sharing that. Appreciate it. Matt or Russ, any other questions you want to add on?
No.
No? All right. Well, Ronald, thank you so much for joining us today. This has been super informative, and I'm sure it will be super helpful to our students as well who are able to watch this recording. So thank you so much for joining us today. We really appreciate it.
Thank you for the opportunity, and
Yeah.
have a good trip, guys.
All right.
Thanks.